So the other significant concept I'd like us to review here is that of receptors. And receptor proteins, are embedded in membranes. Sometimes they're actually intracellular. They're just found inside the cell, actually. So whatever the agonist is that's going to activate them has to cross through the membrane. We talked about ionotropic receptors, right? And we talked about metabotropic receptors. These are also called ligand-gated, ion channels for the ionotropic, because a ligand is a fancy word for chemical, right? Like a neurotransmitter or a drug that'll bind to the receptor. And if it's an ionotropic or an ion channel, right? Ligand-gated ion channel, the binding of the binding of the neurotransmitter will cause it to change its shape and open a channel that will allow for, some kind of an ion to pass through, like sodium, for example, or, potassium or chloride, right? If sodium is allowed to get into a neuron at the resting potential, right, it's going to depolarize it because the sodium is going to be driven in, and it's going to, make it more positive inside versus the out, and it's going to make it, bring it towards the threshold for the initiation of an action potential. If it's potassium, of course, it's found in high concentration on the inside, so it'll exit the cell, right, which will, remove it positively charged ion from the inside and take it out, and that'll, hyperpolarize the cell and make it less likely to, initiate an action potential because you'll be farther away, from the threshold potential of minus 55. If it's chloride, right, of course, the chloride is going to, go from the outside and work its way in because it's driven by concentration gradient force, opposed by the electrical force, but the concentration gradient force wins at, minus 55, 70 at the resting potential. As the chloride comes in, however, it's going to increase the charge difference, make it minus, 75, minus 80, and, you're going to get those forces sort of balanced again, but it's going to be a hyperpolarized, status and, farther away from the threshold potential, and therefore inhibitory and less likely to fire an action potential. So these are ionotropic, right? But remember, there's some complexity here because these ionotropic ones can also, be ligand-gated. They can be gated by voltage, right? The voltage change can, open or close some of these channels, it turns out, and, some of these receptors. Sometimes it can be both ligand and voltage dependent in terms of these receptors. It could be second messenger regulated. So you could actually have the metabotropic receptors. Remember, these are the ones that don't have an ion channel that is attached, or that is part of the actual structure, but they have a G protein attached on the inside. This is the intracellular section here. This is the extracellular section. The G protein will be released upon the binding of a neurotransmitter, and then it can go off and it can bind on the inside to another channel that'll open up and allow for sodium or chloride or potassium to flow, or calcium to flow. It'll alter the membrane potential. So you've got a lot of complexity in these receptor systems that we don't talk about in introductory classes so much. Remember, the terminology is complicated too. You got ionotropic or ligand-gated, right? Is ligand-gated channel. Those are two of the same thing. And then you've got the metabotropic, or the earlier name, which is actually quite commonly used here in the Pacific Northwest and in many research labs, which is the G protein coupled receptors, right, or the GPCRs, right? And I'm indicate on our website here a couple of other kinds as well. There are these tyrosine kinase receptors, receptor tyrosine kinase, high affinity cell surface receptors for many polypeptide growth factors. A lot of hormones bind there, etc. And they transfate or phosphate groups to chemicals on the inside of the membrane. So it's another class of receptors, usually stimulated by estrogen and other hormones, for example. Then there are intracellular hormone receptors. So these are receptors located inside the cell rather than on the membrane itself. So whatever the agonist is, whatever the hormone happens to be, actually has to work its way through the membrane to access it. But it can usually pass right through, to access it. So if it's traveling in the blood, it'll reach those intracellular receptors, which will change their shape, right? And then can then activate or, phosphorylate or, change the structure and thus the function of the next chemical in a biochemical cascade. Remember, ultimately, these biochemical cascades can involve the entry into the nucleus, right, of specific activated, compounds that'll bind to specific regions of the DNA and either promote the transcription, and then the translation of those protein products, or inhibit, right? So it can alter gene expression because of the listening by these receptors and the transfer of that information biochemically back to the nucleus. So my point here is that there's a great deal of complexity in these receptors. They're not so necessarily just driven by one thing or another. There's many different kinds. Here in local campuses, local research labs, there's a lot of study of opioid receptors, for example, and some opioid receptors will, when they are stimulated too rapidly in succession, they'll actually pull themselves into the membrane, so there's no extracellular component at which an opioid, neurotransmitter or an opiate drug can act. We talked about the nicotinic cholinergic receptors, right? If they get stimulated by acetylcholine, too much acetylcholine, too rapidly, they actually shift into a different form that is desensitized, where they no longer respond to the release of acetylcholine, to the stimulation of acetylcholine, which is very dangerous, right? Because the acetylcholine is released in the peripheral nervous system at the neuromuscular junction, and if there's too much stimulation at those receptors, and they shift into forms where they're no longer responsive, well, you're not going to be able to move muscle, and people can suffocate or asphyxiate because you can't move the diaphragm. So receptors are very complex. They're often driven by more than one factor that'll determine whether they, open a channel or release a G protein or phosphorylate something internally or activate a series of biochemical cascades. So very complex.